words, at the end of, let's say, in the late 50s, we could say that you can explain conduction in the peripheral nervous system, you can explain muscle activity, and a range of other similar types of electrical phenomena in nervous systems. But, and that is an amazing achievement, you can also begin to understand how the heart works and how it conducts. Amazing achievements. But really, we don't know how the brain works. We need a different level of information, a different level of understanding about information processing to understand how the brain works. So I will now take you through a little bit of the history of that. So the next challenge is information processing. How does a neural network take in information? That information can be sensory information. It can be your eye seeing a light and then going down an axon and then going to the back of your brain to your occipital cortex. It could be something like that. Or it could be something, how do you work out an equation? How do you form a memory? How do you judge how fast a ball is coming at you? The information processing is key to that. How does that occur? Well, I will presage that by saying we don't know, but this, in the late 50s, some amazing sets of experiments were started, and one of them was by this guy, Professor Hartline from the States, and he used, again, this was a crab, this horseshoe crab, which lives on the east coast of the United States, and there's actually some in Australia, I think, as well, have a very unusual form of eye that's on the side of the shell. And by doing an exquisite set of experiments, he showed basically how that simple eye could detect edges. So in other words, you've got cells which are starting to act as an aggregate to process information. And that was Hartline's contribution. And there's still, again, a set of equations which will allow you to model how a nervous system will take in stimuli, and how it will discriminate things that are important from things that aren't important. So that was a major achievement. The next point I wanted to bring up was, what I see as the next major advance was a link between neural activity and behaviour. Now, this is where we're getting close to something. Now, we're not going to get a link between why you went down to the shop and bought some milk today and your brain state. No, we're not going to get there. We need simpler systems. And this was the work of Eric Kandel. All these people I've spoken about, by the way, have all got Nobel Prizes for this work. And Kandel started working in this, this preparation, which I used to do experiments on actually when I first started my doctorate. It's called aplysia, or the sea hare. And it's basically a sea snail that really doesn't do too much. You wouldn't be impressed by it, but it has a very simple nervous system. A lot of these types of snails, the invertebrate nervous system seem to be divided up into two types. There are the snails, which have neuron systems of, let's say, can be counted in terms of perhaps maybe 100 neurons in their whole nervous system. And there are things like ants and bees, and I know this because I thought, why don't I do some experiments on ants? And the reason is they have lots of nerve cells. There are, I don't know how many, but thousands of them, and they're really small. So that's why I've never worked on ants. But these ones, these sea snails, have these big neurons that are about 0.5 millimetre across, and you can get your electrodes into them, and there aren't many of them. So if this animal does something, and you can record from some of those neurons, you can potentially get a readout of a behavioural state. And this is what Kandel did. He made the animals learn a behaviour, and that was to give them a noxious stimuli, and they developed that behaviour. Then he recorded electrophysiologically and found that he could see changes in the neurons associated with that learnt behaviour. And because of that, he got the Nobel Prize, and rightly so. So we're heading towards somewhere where we might be able to make some higher order sorts of understanding of human behaviour or animal behaviour with these major advances. And all this was in the late 50s. He's still working on this area. He's still extremely productive, and he's made very large inroads into how memory works. And one of the main aspects of this, these experiments was not just a behavioural correlate, but how memory is encoded. So the next thing I wanted to talk about on a similar line, how, again, higher cortical function. How do things happen in terms of you get a peripheral signal coming in, how does it get processed in the brain by many cells? I mean, how is the information getting changed and getting adapted so that it can be perceived? And so again, the late 50s, Hubel and Wiesel in the United States, again, Nobel Prize winners. What did they do? They worked in the cat. Now they've gone from the sea snail, we've gone from the squid axon, now we're going into a mammal. So we're getting a bit closer to where we'd like to be eventually. And what did they do? They chose a simple sensory system in some ways. You might think that the visual system is very complex. It is complex. But your ability, you can put electrodes in the retina, you can put electrodes in the optic nerve and get phenomenal resolution at a single cell level about stimuli in the periphery. It allows you to make very precise observations on how stimuli get encoded into neural signals, and not only that, how they get transformed as they go through different relay sections of the brain. And what they found was that the simple stimuli, like a light in your visual field, will be picked up like that in your retina. But as it goes towards your cortex, those three sets of relay cells down the pathway, the signals that they're picking up are much more complicated. For example, in your occipital cortex, some of these cells will only be activated if they see a bar moving at a particular angle. So the type of sorts of information they are processing is much more complicated than the sort of information that's coming in through the retina. And this may sound, well, yeah, that sounds what I'd expect sort of thing. But it's really telling you something about how the nervous system is working. They developed this idea of the hierarchical processing system, that you had a series of relays where the information processing became more complex. And this, to this day, is actually an area of intense debate. I've worked with some visual physiologists, and I did my first undergraduate project in something like this. And I worked with this older guy who he's not, I don't work with him, but he's in my lab next door. And so he still does these experiments 5 days, day and night, on the cats, because that's how difficult these experiments are. And I say to him, and he's going, oh, Hubel and Wiesel, gee, they were wrong. And he's always trying to disprove them. But that's the mark of an amazing piece of work, that it becomes a paradigm that becomes the mark against which you've got to do the rest of your experiments. Not only that, they did some experiments with this term called plasticity, which I'll come across again in a moment, where they actually made a cat blind just by closing up its eyelid, and found that the neural pathways changed very significantly. So it was the first work, at least as I'm aware, in the mammalian nervous system, showing that you could change a neural system by its inputs. A phenomenally profound observation about complex neural systems, that they are modified by their input. So now what I've talked about is experimental work, how we've done experiments, we've picked up this very incredibly interesting information which is telling us more about the central nervous system. But in addition to science and observations, we must have theoretical frameworks. And there are several that I'd just like to run past you quickly, just for completeness. Hebb's proposal that synapse, when cells fire together, their signals will get augmented. In addition to that, there's the McCulloch-Pitts model, which allowed us to actually see how a set of nerve cells could actually act as a computer, could actually sum things, change them, and actually come out with a signal. So that was an important development. And the final thing I'd just like to touch on for completeness is the so-called Hopfield network, which was developed in the 80s, showing that a set of nerve cells connected more or less the same way as a nervous system could act as a memory. So instead of having a little on-off switch like a computer, where you've got a memory specifically in a very particular point in RAM, this model will give you a memory encoded in a set of neurons, which can change or be modified. So that, again, was a major theoretical development. So now I'd just like to touch again upon this topic of plasticity that we came upon just briefly. The nervous system changes with its inputs. So as we talked about the Hubel and Wiesel experiments with kitten vision, there's another thing that was developed in the 80s called long-term potentiation, where we could actually change the output of a neural system by electrically stimulating it. But The main point I want to take away from that, there's a book published a few years ago called The Brain That Changes Itself. This concept that the brain is continually changing to its external stimuli and perhaps to its internal stimuli is such an important paradigm. I just can't emphasise enough. We are not dealing with what you would regard as a normal computer here. A normal computer is structured to do very particular things in a very particular way. If you change any of it, it breaks down. A neural system is constantly modifying itself, and I don't think we have the faintest idea at the moment how to incorporate this into our understanding of complex neural systems. So I won't, for the sake of time, I won't go into any of these in great detail, but we have a reasonable idea now. I would say we have a reasonable start of understanding how memory might be encoded in a mammalian neural system. We have some interesting data on how motivational states may be encoded in higher centres in the mammalian brain. We have a reasonable idea about how sleep can be induced into a mammal, for example, but I won't go into those in any detail. I just wanted to run past you some of the technical advances that have occurred in the last 20 or 30 years that are really changing the way we can start to analyse these problems. The cellular level patch clamping I won't talk about, but we can now put optical readouts into neural systems and see how whole sets of neurons are firing at the same time. That's been a major change. In addition to that, if we find, for example, that a particular illness, for example, schizophrenia or epilepsy, has a particular genetic defect, we can put that genetic defect into an animal and then study that animal's nervous system intensely to find out how that has happened. And it gives tremendous insights into how the nervous system is working. The other thing you might hear about occasionally, this is the topic of optogenetics, where you can actually you can make the cells in the brain of an animal react to light. Now you may say, why on earth would you do that? Well, it allows you to very specifically activate particular sets of nerve cells. I won't go too much more about that. Now, another area that's been a massive change. Sorry, am I? You can hear? Sorry?
Can I just interrupt?
Yeah.
There's some elderly members of the audience who are not able to hear you.
Sorry.
So if you can speak into the microphone a
Yes.
little bit more.
Sorry, it was ringing a bit. Sorry. So the next thing you will have heard about is obviously MRI scanning. Now, better?
Yes.
Yes, good. MRI scanning is magnetic resonance imaging scanning. Now, you'll be aware of that. It's amazing technology, and I won't talk too much about it, but an even more amazing technology in some ways is the ability to have functional MRI, where we can get you to actually think about something or do an activity, and we can then image that in the brain. Now, in the top diagram here, there is a picture of someone actually encoding it, is memorising something. And those coloured areas are parts of the brain that are actually activated during a memory activity. This lower panel shows you someone engaged in speaking. And as you can see, there are several panels, but we can see the parts of the brain that are activated. The other thing I just wanted to point out was that on the bottom panel you can see what looks like a very colourful set of ribbons. Now that's actually called tractography. We can actually image the actual tracts of where the nerve fibres are connecting to one, or nerve cells are connecting to one another. I just want to say as a little aside, people love these diagrams because they look so pretty. And I often say to my colleagues, why do you do tractography? And they go, oh, it looks so nice. So it's one of those things where we're looking for something to do with. But I just would warn you against the flashy presentation of the neuroscientists sometimes. They try to impress with colour, but it's not always that substantive. I did want to just let you know about something we've been doing at the Royal Melbourne and the Melbourne hospitals. We've been able to implant chronic electrode recordings in humans who have uncontrolled epilepsy, and we've kept them in for about 2 years, and we've been able to predict epilepsy when it's going to happen. Now it's not foolproof, it's not the cure for epilepsy, but it's an amazing technology that we were very privileged to be able to share, to derive from the United States. And that was recently published in Lancet. Now, what I did want to bring up as well was the topic of computational neuroscience. And you will hear more about this I'll just touch upon it, but simply that computing power, computers are becoming so powerful now that there is a sense that we can model large neural systems. We can definitely model large neural systems, but there is a kind of what I think is perhaps an excessive optimism that we can actually realistically model large neural systems. Up the top left there, I've got the IBM Blue Gene system. We're fortunate to have one of these systems in Melbourne. I think I'm not sure how many, it's probably 4 or 6 in the world. And these are these petaflop devices that you can do amazing simulations on. The middle one is just to show you how nice some of the coloured pictures they're deriving in the Blue Brain project are. And then the bottom right-hand corner is a project called the Blue Brain project being run out of Switzerland, where they're attempting to use these supercomputing systems to try to really get into how a whole realistic mammalian nervous system would work. They're hoping to realistically simulate it. I'll come on to that more in detail, but I did want to point out to you this technology is amazing. We in our lab we simulate neural networks for epilepsy, for types of we're looking at phenomena associated with schizophrenia. We can model drug effects and so on. Another thing we are doing is modelling whole proteins. So, for example, a 2,000 kilobase, or sort of 8,000 kilobase protein, in other words, a fairly big one. We can model it at an atomic level. So that means there'll probably be thousands and thousands of atoms, all in space, and we can simulate the activity of that protein, very slowly, I must admit. But that's been an amazing development as well. So a blue sky thing I just wanted to talk about that we are thinking about and may even do something about, and that is that the ultimate thing would be able to get a readout from the brain in real time of the electrical activity. Now we can do that already in the sense that we can put an electrode in. We can put multiple electrodes. That's another technology that we have. In my lab we can put up to 32 electrodes in a brain at the same time. But to be able to, without non-invasively being able to detect electrical activity in the brain at the cellular level is probably, it might not be possible, but using MRI technology it is just feasible. So we are thinking about running some experiments to see if we can pick up a signal from a nerve cell directly using MRI in real time, maybe in the next year or two. So the real question though, so I've talked about where we're at right now is I've tried to tell you how a nervous system might work. I've tried to tell you how certain aspects of higher cortical function might be analysed, might be understood. And I think at this stage there's a whole lot of things I haven't said that time doesn't permit. The understanding of the nervous system with regard to things like depression, with regard to schizophrenia and so forth, we are beginning to have a little bit of a explanatory framework functionally in terms of the central nervous system. So we've come a long way. That's what I wanted to convey to you. The science is amazing, the knowledge is amazing, and everybody in the field is very excited about it. But what I wanted to do now was just briefly touch upon larger questions. So can we explain the human mind with these mechanisms? So I wasn't satisfied with just telling you how the brain works. Now I'll tell you how the mind works, but I want to convey to you that I don't know. So the real question, so free will, moral action and consciousness, can these be derived from these sorts of models? Now I don't know what the opinion in the audience would be, but I think my mother would say no. And one of the sad things, my mother passed away a little while ago, but one of the sad things, not sad, but ironic things of life, isn't it, that everything your mother said actually turned out to be true. And anyway, she wasn't a neuroscientist, she was a very humble stenographer, I think. But she would still say, no, I don't think so. And I'll come to why I think she was right. So let's just, I think I should finish up fairly soon. Yeah. But historically, again, there's a wide range of opinions, which I won't go through in detail here. One thing that I did want to say was that there's a philosopher named Wittgenstein who was in the 20th century, and the thing about him was that I disagree with most of the things he said, but he was such an admirable character. I really like him. And there's this famous quote he has that what we cannot talk about, we must pass over in silence. And the question is, should we, in terms of these topics of consciousness, free will, etc., Should we just say, look, we don't know what they are, let's forget about it, you know? And the trouble is, the answer may be yes, but the trouble is that a lot of people are talking about it. And I did want to bring this up because whilst it's a very intriguing and stimulating topic, there are people in big centres throughout the world, especially in Harvard and the United States centres and actually at Cambridge, who are saying that yes, we can actually deal with these topics in terms of neuroscience. And all I would say at the moment is that I think we're very far from being able to do that. And the other thing I did want to bring up was a concept that in science, it has an enormous explanatory ability. We all know that. The revolution in science has allowed us to sit here, have air conditioning, have projectors and all that sort of stuff, which is amazing and given us much better healthcare. There's no question at all. But there is a kind of, I think, a funny side to science that it sometimes you get a model or you get a system that becomes the reality. There's this funny sort of dialectic tension between observing things and what the model that you make sometimes becomes the reality. And I think in this particular area we need to be a bit careful that the model that we derive somehow becomes the reality. We only see human beings in terms of a set of neurons or a set of mathematical equations, which I think would not only be unfortunate but quite wrong. So just a little history of this topic. Eccles, who I spoke about before, and Popper sought to preserve free will in terms of neural system on the basis of the random nature of synaptic transmission. In the late 20th century, there's a guy named Dennett who's based in the East Coast and States, who thinks everything is based on mechanistic principles, but he really thinks free will is still there, and no matter what. Now, it's a little bit hard to see how we can justify that, but he describes free will as the most difficult and the most important philosophical problem confronting us today. And then I won't read this all, but he really brings out the crux of the matter, that free will has such important implications that if neuroscientists go around saying it doesn't exist, it has a lot of implications. Steven Pinker, again, has various sorts of ideas, which I won't go on to. The Blue Brain Project, this project that's been funded in Europe, predicted that in 10 years they could make a hologram that would be conscious. That was in 2009. And in 2013 they got €1 billion to follow through on this project. And I just had a quote from Christoph Koch. The roundworm has exactly 320 neurons and we still have no idea how this animal works. So I'll leave you to draw your conclusions. And I won't go too much into that. One person I have thought she gave a lecture at the University of Melbourne just recently, I thought had some very sound ideas on this, that the problem of defining consciousness really is extremely difficult, and to say that you're going to develop a system that will replicate it or explain it, I think is maybe outside the scientific scope. It might be, but at the moment it's too difficult to say. So just finally, a final quote from Dennett. The history of philosophy is the history of very tempting mistakes made by very smart people. And if you don't learn that history, you'll make those mistakes again and again and again. One of the ignoble joys of my life is watching very smart scientists just reinvent all the second-rate philosophical ideas because they're very tempting until you pause, take a deep breath and take them apart. So I'll leave you with that. Thank you.
Thank you, sir. Ladies and gentlemen, can we have a standing ovation for our eminent speaker, please? Thank you, ladies and gentlemen. I now invite Dr. E.S. Krishnamurthy to moderate the question and answer session.
We can take 2 or 3 questions if any of you have burning questions you'd like to ask the speaker. Dr. Ramaswamy, another hand going there. Yogini.
It's a real fundamental question. We say control your mind, control your mind, control your thoughts and all that. And Mr. Murari was referring to a person who was shooting, imagination, running riot and so on. Now, the point is, we know that all the brain activity is electrical. Now a thought arises. See, you can record it with PET. The neurons show reaction in color. But then, what did the neuron start the thought, or the neuron followed the thought? It's a very difficult thing to answer. And if we are able to say, you control the mind, and thought, they will say, who is that you and I? If I'm able to get at that I should be very happy. So yogis do that. So I'm talking from my experience of hypnotherapy. We do so many things in hypnotherapy. In fact, we show a person a blue pen and then say, oh, how beautiful the red color is. The person sees only the red color. What the mind does not see, the eye does not see. So, and then Descartes said, I think, therefore I am. But that means I am a slave to my thoughts. Whereas I say, I am, therefore I think. My thoughts are under my control. And who is that I don't know.
Dr. French.
Well, that's, as you said, that is a difficult question. So all I would say is what my mother would say is that I think that there's definitely we really do experience free will as a subjective phenomenon, and that so the neurons are probably not what is driving it. That would be my take on it. But it is very difficult to give a definitive explanation.
Yes?
Yes.
Good evening. I'm Anandi Karthik. I'm a counselor. Let me first thank you for the wonderful presentation. Out of my free will, I'm just asking you this question about today with the gadgets. How does it affect the brain? Because the younger children are more exposed today. All the gadgets and the TV and the media, everything is exposed to them. How does the exposure affect the brain?
Look, it's such an important question, and it's way outside my field. But I was reading there was a lot of thought that the kind of stimuli that are going on must be having an effect. And I was just reading a report last night that we can do measurements of connectivity in the brain using functional MRI.
Might want to use this one.
So we can do, there are new types of, using functional MRI that I was talking about, we can actually look at how the networks in the brain are actually interconnected. Now, what that actually means is difficult to interpret, but I was reading a study last night that suggested that people who had been excessively exposed to gadgets or particular computer games, etc., or had developed an excessive interest in that, had changed the connectivity in their brain. Now, it's a one paper, it's a technique very difficult to interpret, that's all about it. But it's just that kind of information that makes you think maybe something could be going on. That's all I would say. But it has to be done at a qualitative level, I think, to start with.
Is there any particular study? How does it change and what impact it gives?
Look, I can't answer that. I'm sure these things are actually of massive interest, and there are a lot of studies. I would be amazed if they weren't happening right now. It's just not an area that I'm really across. But I think a Google search or a PubMed search will bring you up many publications on that.
Good evening, Dr. French. My name is Yogini. I am a doctoral student at McGill University in Montreal.
Yep.
First of all, I've really enjoyed your lecture. So my question is fairly general, and I'd like to ask you for your perspective as a neuroscientist. And I think I'm asking you this question because it has a lot of implications for people who are working with mental health, mental disorders in the Indian cultural setting. So my question is that as a neuroscientist, from a neuropsychiatric perspective, what do you think is the role of other emerging disciplines that look at the non-biological, non-brain-based basis of mental illness and mental disorders? So sort of the cultural and social factors in the environment that affect the etiology or the treatment of mental illness. What do you think, or do you think there can be a reconciliation of the neuropsychiatric perspective and then the cultural, social, psychiatric, or other models of mental illness
Yeah.
and mental health?
I mean, Krish would have be the best, by far the best answer on this. He's right behind. But look, my take on that is a very interesting point, because on the one hand, I can say to you that the neuroscience of schizophrenia and psychotic episodes is going ahead. Well, I'm doing some work on it myself. And I would envisage, this is where we have to get the right balance. I anticipate that we will make discoveries in terms of autism, in terms of schizophrenia, that will allow us to change therapy, improve therapy. But on the other hand, there's this really important dimension of the phenomenological dimension. What is a human being? The human being is in their social situation, they have needs, they have supports, and I couldn't support more the need for a multidisciplinary approach to these problems and enculturation. And we experience this ourselves in our psych units and so forth, that they become way too organically based. You treat with a drug. I had a case recently of a friend of mine. His son was having psychotic episodes, had been treated with basic conventional antipsychotics, had a little bit of improvement. But what he needed was a much broader approach, that Krish has been talking about. And he got much better with that. And with a little bit of intelligent tweaking of the drugs, he got much, much better. So look, I couldn't support more of those kinds of approaches. I think they're very important.
So you think there is a reconciliation and there can be a way to bring these two somewhat opposing perspectives?
Well, I think also, I do engage a fair bit with the psychiatrists, and a lot of my patients have issues. But so part of it will be in the cultural situation of also education, of course, because people, I imagine, in where I come from in Australia, don't understand psychiatric illness, what it really is, and people can be ostracised, or they can be treated in a peculiar fashion. But to give the family and the culture in which they're in sufficient information, what's going on, and use those supports is the ideal situation. I think it has to cut both ways. That that they're brought in a constructive and synergistic way.
Okay. I'm sorry, just one more point. Can I make one more point? I just wanted to say specifically, like suicide is a very classic example where a lot of social suffering can lead to what seems to be almost like an insane act to take your own life.
Yeah.
And yet there's also a huge amount of research that would suggest that there's some genetic component, there's some brain-based component to, this particular act. So especially in India, where social adversity, economic crises, things like that lead to a lot of mental health problems, and so sometimes even impact the disease progression or the severity of the disease. So I just wanted to know if you felt that whether the social and cultural context has an impact at a brain level on that synaptic plasticity and all of that you were talking about. I'm just going back to Kandel's famous paper where he talked about psychoanalysis and brain biology.
The answer is that yes, I think it does. And the other thing that's developed more recently, I didn't touch upon it at all, is epigenetics, and that we are finding immense. It's almost gone back to a Lamarckian model where your environment is affecting your genes. So I think all these things play into it, but I can't help but that the social interaction is incredibly important with these patients.
If I may just add to what was being said, the hippocampus, which is an incredibly small structure the size of a finger joint, is now being blamed for most things that we suffer from. It's blamed for epilepsy for many years, now blamed for major depression and schizophrenia. And I think a lot of the research is coming from Melbourne, actually, where first episode psychosis shows a reduction in the size of the hippocampus, and repeated episodes the hippocampus becomes smaller and smaller and smaller. So how this small part of the brain has so much to do with human emotion is something which is hard to fathom. I'm going to take two more questions. I see Mr. Srinivasan's hand going up and Dr. Kamakshi's hand going up. In the end, sir.
Thank you very much for an excellent lecture, and thank Krish and my teacher, Professor Srinivasan, for organizing such a beautiful lecture. Just comment come questions. The left hemisphere and the right hemisphere are not truly symmetrical.
No.
There are some hemisphere asymmetries. Like the left occipital horn is larger, the left temporal lobe, there are differences. And it is stated that in developmental dyslexia, these hemisphere asymmetries are not present and may be causative. That's one of the theories offered. Would you subscribe to this theory?
Not without further information. But look, I think the answer to that, with these, one of the very powerful tools that we now have is the volumetry for using MRI. I myself don't use it, but many of my colleagues do. So, for example, in schizophrenia or various types of mental disorders, we can find subtle volume changes and so forth. So in this case, you're saying that they're actually more symmetrical, that volume changes are not there. So my point is that as an explanatory framework, we need to be careful not to go, oh, despite what Krish is saying, the hippocampus is bigger or smaller. That's a very important clue and needs to be followed through. But you need to go much further into that. So whether the reason why, even if I actually was up on the topic more than I am, I don't think the best person in the world could actually give you a real answer to that question, because we just do not know the mechanisms that well. That would be my take.
Thank you very much.
Two final questions.
Only one more. This is about legal implications for a physical basis of behavior. Say a person commits murder, is a psychopath. Will he escape punishment like capital punishment just because a piece of the brain, like the amygdaloid nucleus or whatever it is deficient? Can it be a justification for his escaping punishment?
It's an interesting story. In Australia, we don't have the death penalty, and it was stopped in about 1966, I think. But some of my older colleagues tell me that anyone who committed murder would almost certainly get a diagnosis of epilepsy because it would get them off on manslaughter. And that's true. I mean, if you actually did have an epileptic fit, you would be in a situation where you were not acting reasonably or rationally. But the point is, this is the point that's so hard to define, that if you take that this is the point that Dene is making, that if everything is taken to be purely a mechanistic function, you really cannot ascribe guilt to anyone that if you do not have free will. And that's what, all I would say is it's a critical question. It's not a trivial question, a critical question.
I have one question.
Yes.
Dr. French, during your lecture, you talked about the scientific investigation of concepts like free will and things like that. So basically, science is attempting to investigate the concepts of philosophy which we have been taking as granted on that. I think there's a third dimension which exists in India, which unfortunately has not been tapped. And that is the enormous amount of knowledge that we have in our Vedic literature. It is our belief that what is not there in the Vedas is not worth knowing on this. So really, not only we take absolute as what is mentioned in the Vedas, but there are branches of Vedas which give the, if I may use the word, scientific explanation for these kind of beliefs. Unfortunately, most researchers have not tapped into this reservoir which exists, which may, in many aspects, facilitate the understanding through scientific investigative tools. So when you are spending €1 billion to do, what was that, the Blue Brain project, but perhaps a little part of that, if it is used by real seekers, not people who are trying to expose what they think is not true, but if they are really in the real seekers of knowledge, even they could be doctoral students I think you will find many of our Vedic centers would be quite willing to share what there is knowledge. And it will be an important work for these seekers to interpret and translate it into a language which all of us can understand, or the medical fraternity to understand. So it is only a comment, I'm not making a question, but to draw your attention, I'm sure many of you know about this, and perhaps what we see today in TriMed, when they're using this integrative medical therapy, is an acceptance that it is not the allopathic system alone which works. And many of the things that they are doing today, I'm sure Dr. Krishna himself does not understand. But, but he's doing it on that. Thank you.
I'd just briefly say, look, the main point I think we completely agree, I will actually look into that, but the science isn't a standalone, it isn't a self-sufficient enterprise. It must stand within a setting of wisdom and a certain set of expectations of the world, which are often unacknowledged. And I think that's something we really need to reflect on.
If I may very quickly respond to the question about the psychopath. The ongoing research by Blair and his group in UCL, University College London, actually shows that people with psychopathy have difficulty with recognizing faces and facial emotions. So one of the reasons why the psychopath beats you until you're dead and not just until you're frightened or until you retreat is because he does not see the fear in your face. That could well be a legal defense in the future. So, and the other thing is that the amygdala actually is larger in people who have emotional discontrol. That's again an evolving literature. So you have a hippocampus that's smaller and an amygdala that's larger. And I would say watch this space. You never know. I'm going to request Professor Rajan, who's our faculty advisor, to ask the last question, and with that we will close.
Thank you very much. I'm extremely happy to listen to a wonderful lecture. I'm a physician. I retired as head of the Department of Medicine, Stanley, in 2003. One of the brilliant patient asked me, "Doctor, you are a senior physician. Can you tell me where the mind is located?" Number one. And he said, I read, mind is constant from cradle to graveyard. Body with us. If that is the case, brain is withering. As age advances, brain functions deteriorates. If that is the case, mind is constant. So the interrelation of these things, there is no deterioration of mind, then where is the mind, he was asking. So incidentally, I watched this, memory is directly proportional to the hippocampus size. Increase in size, more memory. Shrinking, loss of memory. But I'm surprised, some of the schizophrenics have got excellent memory and they get distinctions, awards, medals and all. How is that? Can you answer?
Take that?
As a medical student, I got into trouble a few times by saying, I don't know. But I suppose it's all, that's the essential question, is how do you reconcile the structural explanations with the phenomena of the mind? We don't know.
Maybe you can look at the Vedas.
Indeed.
Thank you very much, ladies and gentlemen. Thank you very much. We now request Dr. Rema Raghu to deliver the vote of thanks.
Distinguished guests, ladies and gentlemen, on behalf of NeuroKrish and TriMed, I have the privilege of proposing the vote of thanks this evening. I would first like to thank our esteemed guest, Mr. Murari, for gracing the occasion with his august presence. It was an honor having you with us, sir, and for listening to you too. I express my deep gratitude to Dr. Chris French for his wonderful lecture this afternoon. Thank you very much, sir. I would like to thank our mentors from the medical fraternity, Professor Krishnamurthy Srinivasan, Many of us at TriMed have had the privilege of having trained under him for many years, and we miss the good times that we had at BHS. Mr. Srinivasan Acharya, thank you very much, sir. And Mr. V. Srinivasan for their support and for their encouragement in all our endeavors. Thank you very much. And I thank the Savera Hotel management and staff for their wonderful hospitality, for the arrangement and for the food. And thanks to Vision Hire for the audiovisual services, the Good Impressions for their printing services, and Marundeshwara Enterprises for having organized this event so well. And a big thank you to this wonderful audience. Some of you have been with us since 2:30, have listened to us, asked us questions. Thank you very much. And I think it's a good opportunity to thank Dr. ESK. He's a cerebral stimulant of our team. Each time he's back from a conference, he has these recurrent eureka moments, and he's up with these new ideas. And then we all try hard to keep pace with the ideas. So there are new programs, there are new orations, and we are preparing right round the year. And that keeps us on our feet. And once again, a big thank you from NeuroKrish and TriMed for your blessings, wishes, and support in all our endeavors. And we're looking forward to have you tomorrow at Velacheri, where we'll be inaugurating our new center. And Dr. Chris French will be with us tomorrow evening. Thank you very much once again.
Ladies and gentlemen, please rise for the national anthem.
Thank you. I hope it was okay. Thank you.